Okay, everybody, hello. Can you hear me? Can you see me? Six students right now are connected. Can you hear me? Can you see me? Please type on the right panel. Ah, yes, okay. Okay, thank you. Thank you. So, I want to discuss today some biochemical problems. Not too much, but I think we will solve them. All right. First topic. The topic name is biological membranes. So, I already published links on lectures. This one is my lecture, biological membranes. These are cell membrane overview, cell-cell interactions, transport across cell membrane is American Khan Academy. I strongly recommend you to listen these lectures, to view, to see this, to look these lectures. So today, here you can also download my presentations. This one, biological membranes, the first one, and this second lecture, this one. You can download the second part of these lectures. So you should to read a topic, Lieberman. I don't remember 10th and 11th topics. Should wait a second. What? Here. Yes. A relationship between cell biology and biochemistry, the structure of membranes, cell signaling and chemical messages, mechanism of transfer of signal molecules between cells, and activation specific biochemical routes. So the next point, what is it? Plans, self-control questions. You also may read them. These questions, biological, according to the structure of and definition of biological membranes. These are. And the last one is homework. I already downloaded. Wait a second. These are homework problems. So we will discuss right now, and after that you should to pass to this test, like homework, the homework and classwork, because you are in your countries, so these are the same quiz, computer-based test. And now we will discuss Mohammed, good morning. Yeah, I see. All right. Right now we will discuss two answers on biochemical problems, and after that you may ask me. You can ask me right now if you don't understand something. You can type right now your questions, and after discussion of these problems, I will answer for your questions, and that will be. And after that, you have to pass computer-based tests, 2 computer-based tests. So first, homework, and classwork. All right, let's discuss. So I want to see. Can you see me or not? Ah, it's okay. You can see this, the text. And this dark text, and I see your questions if you have ones. All right, the first problem, this one, 3.1.3.1. Many growth factors upon binding up to the receptor exhibit downregulation in which the receptor number on the cell surface decreases. This occurs due to which one of the following process? Explain your answers. What does it mean? The first question, what is groove factor? Groove factor, you can look in Wikipedia. What is it? What is it, groove factor? Wiki. Groove factor, English version. A groove factor is a naturally occurring substance capable to stimulate cell proliferation, wood healing, and occasionally cell differentiation. So, in simple words, what is groove factor? Groove means increasing of number of cells or size of the cell. And groove factor works like a signal molecules which stimulate proliferation or division of cells, wood healing or migration of different cells into the wound, and cellular differentiation is a conversion one type cell to another cell. Okay, the next question. They are binding to the receptors because to activate some specific properties of this groove factor, these groove factors have to interact with surface molecules. We call them receptors, and after that, inside the cell, they activate some specific biochemical process. The question, many groove factors upon binding to the receptors exhibit downregulation in which the receptor number on the cell decreases. What does it mean? That means that after activation specific biochemical process inside the cell, to prevent overfunction of this biochemical process, number of receptors on the cell surface decreases. That's it. Okay, the next question. Problem 3.1.3.2. The regulation of heterotrymeric G proteins is similar to the regulation of which one of the following processes? Explain your answer. Regulation is similar to the regulation of which the following. Active transport, facilitated transport, regulation of heterotrymeric, similar to regulation. So, what does it mean? So, what is heterotrymeric G proteins? Let's look. These are heterotrymeric G protein. I want to look at the membrane. This one. Heterotrymeric contains three components. The first one, G alpha subunit, G beta subunit, and G gamma subunit. So we call these molecules heterotrymeric. That means they have three subunits, trimeric. And hetero means different, alpha, beta, and gamma. So the next question, regulation of which one of the following process? So the regulation of these proteins. Regulation is a movement inside the membrane. They don't change activity of DNA synthesis or RNA synthesis, or they very similar to active or facilitated transport. I prefer facilitated. Facilitative active transport is using of energy. According to the structure, according to the mechanism of action of these proteins, look at this. They use GTP, hydrolysis, is very similar to active transport. Active. So they use these heterotrymeric proteins use GTP as a source of energy. So the true answer is D. D. All right. All right. The next question. The next question. Problem 3133. The release of insulin from beta cells of the pancreas requires calcium ions influx through the channel that is activated by a change in the membrane potential across the plasma membrane. The movement of calcium across the membrane is an example of which one of the following. Explain the answer. Okay, so let's look at the mechanism secretion of insulin. Secretion of insulin. Regulation of insulin. Insulin molecule synthesis, translation, translocation. What is it? Ribosome, no. Releasing. No. Glucose, glycogen, receptor. Mechanism function. Insulin. So insulin action. Mechanism of action. No picture. I want to show you another picture. Lecture biochemistry, biological membranes, amino acids. I want to show how does it work. I want to show you here in clinical biochemistry. I have a picture. Clinical biochemistry. Carbohydrates here. I want to show this slide. How does it work? Relationship between here. Yes. How to increase. Let's try. Let's show you. So can you see or not? Can you see? Yes, I think you see. You can see. So instead of we have G proteins, I don't know, Hetera G proteins. Okay, I want to explain how this insulin is secreted by beta cells. All right, so increasing concentration of glucose uses specific glucose transporter GLUT2 in pancreatic beta cells, and glucose passes into the beta cell. After conversion of energy, it works as a result of production of increasing concentration of ATP here. ATP blocks movement of potassium ions from beta cells into outer space. So this protein called ATP gated potassium ions, and the result is opening of calcium voltage dependent channel here. So this is passive transport of calcium from blood into the beta cell, and after that increasing concentration of calcium inside the beta cell, increasing fusion of insulin granules with plasma membrane, and as a result is a insulin secretion. Again, the release of insulin from beta cells of the pancreas requires calcium influx through the channel that is activated by a change in the membrane potential across the plasma membrane. Okay, the movement of calcium across the membrane is an example of which of the following. Explain your answer. Voltage gated panel or channel. Yeah. Voltage gated channel. That's it. The true answer is A. The next question. Transmembrane proteins can best be described by which are one of the following. Explain your answer. Transmembrane. Transmembrane means they have a extracellular domain and intracellular domain and intermembrane domain. So, according to this, according to the structure of transmembrane protein, I want to show you how do they look like. Transmembrane protein. Here. So this is yellow is a membrane. This is extracellular domain, intracellular domain, and intermembrane and membrane domain. Here the same idea. 1, 2, 3 different proteins. So they can usually be dissociated from membranes without disrupting of lipid. No, that's wrong. Because they interact with membrane. So they have two domains, two charged domains inside the cell and in extracellular space. The next question, the next answer. They are classified as a peripheral membrane person. No. They intermembrane. They contain hydrophobic amino acid residues in their carboxylic terminals. No. Because hydrophobic amino acids have to interact with residues of fatty acids inside the membrane. D. No. So A, wrong. B, wrong. C, wrong. They contain hydrophobic amino acids residues extending into the lipid bilayer. Yes, into. They have to interact with fatty acids residues. They contain membrane-spanning regions that are alpha helices. It's very possible. So the true answers are D and E. Okay. Так, so instead of have control plus no. Так, let's go ahead. The next problem. Problem 3.2.3.5. Which of the following is characteristic of the plasma membrane? Characteristic of plasma membrane. Explain your answer. Characteristics of plasma membrane. It is composite principally of triacylglycerols and cholesterol. No. Principally, membrane contain cholesterol or cholesterol esters and phospholipids. B. So answer A is not exactly true. Partially true. B. It contains principally non-polar lipids. No. They contains polar lipids. Phospholipids. They works as a. They have hydrophobic head containing a phosphate group and two hydrophobic tails. So they works as components of membrane. So this is B is wrong. C. It contains phospholipids with their acyl groups extending into the cytosol. No. Inside the membrane. Wrong. D. It contains more phosphatidylserine in the inner than outer leaflet. Possible. It contains oligosaccharide sandwiches between two inner and outer leaflets. Yes, oligosaccharides presumably work. We can find in outer space. I think the most true answer is a D. The A answer contains only partially information, true information. Oligosaccharides outer leaflets. Outer. So the most relevant answer is D. This one. So no problems yet. Yeah. Okay. So this video will be stored on the YouTube, so you can return to this video for preparation of colloquium, and relook these answers. I think I'll help you. If you have any questions about biological membranes, please type me right now. I want to recall you that you have to pass 2 computer-based tests. These are computer-based test homework and the computer-based test class work, and that's it. So if you have any questions right now, please ask me. If you don't have any questions, we can finish right now. Please look again. Presentations, video lectures, textbook, and if you have any questions for next week, you can ask me. Right? Okay. Today we're seven students. Very good. So good. Now is 8. Now is 8. Yes. So we can finish. If you have any questions, you can ask me. If you have no questions, thank you for listening, and we will finish right now. Thank you. Bye.